Hi, and welcome to the AP Biology Lab 1 walkthrough. This is on diffusion and osmosis. You may be watching this at the beginning of the year as you get ready for the lab, or you may be watching it at the end of the year as you review and go over the things you need for the AP test. This is about diffusion and osmosis, and the actual lab is broken down into two parts. First of all, we should talk about what diffusion is. Diffusion is simply random movement of objects or molecules from an area of high concentration to an area of low concentration. So in this diagram right here, this bottle of perfume, if I take the cap off of it, that perfume is just going to spread out. Or if I take this, we've got some air freshener, and I just spray it into the air, those molecules are going to move around, they're going to bump around, and they're eventually going to spread into every area. Like this, I can already smell it, but it's going to spread around this whole room. And that's diffusion. Now, osmosis is a certain type of diffusion, and that's diffusion of water across a semi-permeable membrane. And so in this lab, we do two things. First of all, we look at diffusion, and then we do what's called the potato lab, where we're going to study changes in potato mass over time, and that's due to just osmosis. Okay, so again, to make those two things clear, I think these diagrams are pretty good. Here we've got some solutes mixed in with water, so you can think of this as water, and this is salty water. And so if the salt or the solutes can move through, over time they're going to move out until it's evenly distributed on either side. Now, before it actually goes, we would refer to this side as being hypotonic, or lower salt concentrations, this being hypertonic. And then after we let it for a run a while, it's going to be isotonic. Now, it doesn't mean that solutes aren't moving back and forth, or water isn't moving back and forth, but we've reached kind of an equilibrium at this point. Now, this diagram looks way different than the other one. So this diagram over here, what we have is two different concentrations, or solute concentrations, on either side of a membrane. But in this case, the solutes can't move through, but the water can. And so right here we have water moving through that semi-permeable membrane, and you can see that the water is actually moving through until the concentration on either side is the same. And so that's osmosis. And it would seem mysterious as you watch it to see one side just magically rise up, because you can't really see the solutes. We use this in reverse. It's called reverse osmosis, to actually filter the water that you have in your maybe bottled water. Okay, now the two parts of this lab are pretty simple. The first part is diffusion. In diffusion, what we do is you'll have to imagine a beaker like this, but this is before the actual experiment. Inside this beaker, inside this cup, we've got water, and also inside that water we have a solution of IKI, which is essentially has iodine in it. So it's a chemical that has iodine. And so to begin with, you've got a little bit of IKI, you've got water, and it looks this color. And so it looks kind of this yellowish kind of a color. In the bag to start with, we have water. We also have glucose. Glucose, remember, is a monosaccharide, and we make sure that we know it's there by testing it with something called test tape, and it shows the presence of glucose. It's a way that we can actually diagnose diabetes. And then we also test outside, and we can show that there's no glucose before the experiment. And then the last thing we put is starch inside the bag. Now, starch inside the bag is a polysaccharide. That means it's a bunch of sugar molecules attached together, so it's a massive macromolecule. So then we just let it sit. So I usually start this at the beginning of class. 20 minutes later, we look at it, and it looks like this. Now, the one thing I should have said a second ago is that you ever have starch and IKI, or iodine and starch, in the same area at the same time, they make this bluish kind of a color, dark bluish color. And so after we let it sit for 20 minutes, we see it look like this. And so the kids are amazed because it magically turns from clear to kind of this purple color. What does that imply? Well, it implies that the IKI is making it into the bag. When we test it at the end, we find glucose outside the bag. So that means glucose is actually moving out, the IK is moving in, but the starch is not able to move out. Otherwise, we would have had this blue color out here. And so the pores inside this membrane are permeable to things like water and glucose and IKI, but not things that are much larger than that. For example, it'd be starch, and so that's not able to get out. The potato lab, what we do is we core potatoes and put different sections of the potatoes in different concentrations of sugar water. And so we put them in distilled water, 0.2, 0.4, 0.6, 0.8, and 1 molar, you can see right here. We get the initial mass of the potato cores, and then we get the final mass of the potato cores. From that, we look at their percent mass change. And the reason we use percent mass change is they all, you can see, start with different initial masses. And so you can see that when we put those potatoes in a hypotonic solution, in other words, distilled water, they actually gain mass. That means that they're swelling up. Why is that? It's because water is moving through osmosis into the actual potato. As it gets sugary and sugarier, then we find that the potatoes are actually shriveling up. What does that mean? It means that water is moving outside those potato cores. And so if we graph it, this is the graph, our independent variable then is the sugar concentration, our dependent variable is that percent change in mass. And so you can see as we increase the solute concentrations in each of those beaker, we see a decrease in that mass. In other words, they shrivel up. Eventually, on either side of this graph, that cell wall actually acts as a constraint. In other words, it can't get too big because the cell wall is going to hold it in there, and it can't shrivel up as well because the cell wall is going to hold it in place. And so that's the osmosis lab, and we can look at that slope of that line is going to tell us the change in the percent mass related to solutes in this case, since we're not measuring time. And then the last thing I want you to make sure you understand is the idea of water potential. Water potential is symbol psi, or psi is the symbol for that, and the way I remember that is it's like Poseidon. And so what psi is simply a measure of how much water wants to go from one area to another. And so inside distilled water that's just sitting there, we have a psi value of 0. But if water wants to move into something, then the water potential inside that something is a negative value. And so if we look at a regular plant cell, you're going to have a negative psi value on the inside, so water is going to flow in. As water flows in, that's going to start to swell up until eventually the cell wall itself exerts a pressure back. And so what do you need to remember? Well, there's a few things you could remember. Water is always going to move from an area of high water concentration to low water concentration. You could also say that water is always going to move from a hypotonic to a hypertonic solution. Or now that we know what water potential is, you could say that water is always going to move towards a more negative or a lower water potential. So I hope that helps.